Lunar New Year. Today's the first day for me, Chinese New Year. So yeah, delighted to be able to be here and share. For those of you guys who don't know me, my name is Jason Yang. I'm an assistant professor here in the MB&G department, as well as the Center for Emerging Pathogens. And what I'm going to be telling you about today is some of our what I'm going to be sharing with you about today is some of the recent work in our lab at exploring how bacterial metabolism and gene regulation / stress response pathways regulate regulate drug responses, antibiotic susceptibility responses in tuberculosis, microbiology tuberculosis. And just to give a little bit of context.
Sorry.
So we're good there? Okay. You know what? I'm just gonna let me just use my Let me do this now. Okay, so is everything working?
Yeah.
Okay, great. So just to give a little bit of context. So our overall research program follows a schema. As a lab, we're interested in doing translational research, and most of our projects use a combination of high-throughput experimental assays, along with predictive network modeling, some mathematical models, machine learning to help us understand, identify biological mechanisms that may regulate the phenotypes and responses that we care about. And in our case for today, what I'm going to be telling you about is a story or a couple stories at using this approach to understand drug susceptibility in MTB. So I know that we frequently talk about this here in this audience, but for those of you guys who are online who might be just joining, tuberculosis is a really, really important problem. This past year, actually, it retook like the greatest source of global mortality and morbidity on the world from COVID-19. And today, the way that we treat tuberculosis is really a combination of different drugs. In the case of kind of drug susceptible TB, TB is treated using a combination of 4 drugs: isoniazid or rifampicin, methamphenyl, and pyrazinamide. And over the past several decades, one of the things that's begun to emerge is multi-drug resistance, where tuberculosis, through our extensive clinical use of drugs like isoniazid or rifampicin, has evolved resistance to these 2 drugs. And this is a major problem because it threatens control of global control of TB. And as a result, there's been a tremendous amount of effort in recent decades to develop new molecules, new drugs effective against multi-drug resistant TB. And in particular, for this first story, I'm going to be telling you a little bit about one of these drugs, bedaquiline, which is an ATP synthase inhibitor that very recently was approved by the WHO to be kind of a foundational cornerstone of a lot of MDR TB regimen. And mechanistically, what you should know about bedaquiline is that its primary target, as far as we understand, is ATP synthase. So this is a molecule that is really kind of wrecking the ability of TB to make ATP. And one of the other things to know about Mycobacterium tuberculosis is that it's an obligate anaerobe. So the only way that I can make ATP is through oxidative phosphorylation and through, in general, through ATP synthase. So when we knock down ATP synthase using bedaquiline, we're really killing cells by not allowing them to be able to make ATP. I'm going to give you a little bit of primer on bacterial physiology, which I think is important for the rest of this talk. So, one of the things that's special about bacteria, prokaryotes, a little bit different than eukaryotes, like some of us may study, is that gene regulation.
Jason, so TB cannot make ATP by glycolysis?
Yes.
Okay. Okay, so yeah, through like substrate level phosphorylation.
So did bedaquiline really kill the TB, or was it
So for those of us online, if you can't hear the question, so David Dunow asked, can actually MTB make ATP through substrate level phosphorylation in a pathway of outside oxidative phosphorylation? So I misspoke, and the answer is yes. However, the follow-up question is, does bedaquiline actually kill TB? And the answer is also yes. And I think we're going to be able to maybe share some of why during the middle of this talk. But what I'm going to do now is give a little bit of primer about bacterial physiology, because I think that's important. And I don't think everyone here works on bacteria. So one of the things that's special about prokaryotes is that gene regulation prokaryotes is very different than eukaryotes, and also this genomic structure of the genes that are transcribed is also very different. And so unlike eukaryotes, where you may have genes that are spread throughout multiple chromosomes and they splice together, bacteria have none of that. And typically what happens is that genes that have related function in bacteria reside together, kind of in tandem in something that we call a regulon. And so in the case of bacteria, whose genomes are oftentimes on the order of megabases, and we're talking like maybe 3 to upwards like 10 megabases. You have a bunch, bacterial cells that have a bunch of independently transcribed units of regulons that are, in general, most of these are going to be inducible. And they're going to be inducible, by different transcriptional programs, different sensing programs that allow bacteria to sense extracellular stresses and intracellular stresses. And so today, when we're talking about antibiotics, what I'm also going to be telling you about is how these stress response pathways regulate, TB's response to either an antibiotic stress or other kinds of environmental stressors. And in the case of stress responses, there's two ways that we can think about bacterial stress responses. One is that you can think about stress responses that are highly specific and really get induced only by the specific, whatever molecule or whatever environment is being sensed, as well as general stress responses that respond very generally to several different stresses, kind of illustrated here, and will change bacterial physiology in a way where the cells become kind of pan protected or tolerant across multiple stresses. One of the phenotypes associated with activation of these general stress responses are changes in bacterial metabolism. And one of the reasons why this is something that's become very interesting, especially in the area of studying, say, antibiotic efficacy, antibiotic susceptibility, resistance tolerance, is that metabolism can be a very strong regulator. And one of these mechanisms by which bacteria become pan protected across many different stresses. And so specifically in the case of antibiotics, one of the reasons why this is that antibiotics in general target processes that are involved or necessary for bacterial cell replication. And so if a cell becomes metabolically dormant, if it decreases metabolic activity, then a lot of those processes are no longer being very active. Right? So you have a process not very active to begin with, and then you hit it with the drug. And it's not going to have as much effect on the physiology or kind of outcome of the cell. And so, when we talk about antibiotics, there's really two classes. You have antibiotics that are what are called bactericidal. And so these are agents like bedaquiline, where when a bacterial cell sees this agent, they will eventually die, or the susceptible fraction of those cells will die. And then, bacteriostatic antibiotics or drugs are ones where when bacteria encounters one of these molecules, it basically just stops growing. But then once that antibiotic or stressful agent disappears, and it's able to resume growth. And metabolism, is shown to be really important, at least for at least part of the cidal activity of bactericidal antibiotics. So, changes in metabolism themselves can participate in the killing mechanism of stressful agents like antibiotics. And this is something generally that is a major focus of the lab, something that we've worked on as a lot. And the story that I'm not going to be able to share with you today, Barry Lee, who's an MD PhD student in my lab, has been working really hard at understanding how changes in metabolism and changes in kind of metabolic demand and load can regulate both antibiotic resistance, like a formal genomic sense, as well as phenotypic forms of resistance. But instead, what I'm gonna do is I'm gonna tell you about two stories that we've been working on recently. One of these was recently published. And in this first story, which was led by Nana Fori-Anina, a postdoc in the lab, what we've done is we've focused on studying mechanisms related to how multi-drug resistant MTB become sensitized to this bactericidal agent, bedaquiline. And so, where this project began is, we know that bedaquiline is one of these cornerstone antibiotics that's used to treat MDR-TB today. And, when we were kind of looking at literature and trying to learn a little bit more about MDR-TB, one of the things that we noticed when we did analyses on some public drug susceptibility data sets is that, there's a tendency for multi-drug resistant TB to be sensitized to bedaquiline. So in this experiment, what I'm showing you is, a histogram of about 12,000 MTB clinical strains, where this consortia that was supported by the WHO had performed drug susceptibility testing to about a dozen TB antibiotics. The way you should be looking at this plot is that the x-axis is the drug concentration, for growth inhibition, that's called the MIC, and the counts are how many of these clinical strains have higher or lower drug susceptibility. And what you can see here in orange is that drugs which, or clinical strains which are defined as multi-drug resistant have a tendency to have lower MICs to bedaquiline than the ones which are not multi-drug resistant. And what this means is that these orange strains in general are more susceptible because, again, IC50, IC90 is much lower.
Just to understand, the orange and the plots are the average MIC of a whole bunch of different strains?
No, so these are histograms. So every strain is going to have
What do you mean they're not listening?
normalized histograms. These are normalized counts. There is, okay, so the histogram, right, for each bin, which in this case is actually the MIC, you get accounts for how many, instances of that trait. What we did was we separated the multi-drug resistant from the non-multi-drug resistant. We counted at each bin, and then we normalized it by the peak, because there's a different number of multi-drug resistant versus non-multi-drug resistant clinical isolates in the collection. So this is a normalized histogram. I just spoke a little bit.
So it's counts of survivors or kills?
These are counts of the MIC, of the minimal inhibitory concentration, which is, I guess, is the minimum concentration of a drug that prevents that bacteria from being able to grow. And they can prevent it from growing either by killing the bacterial cell or just halting growth.
Non-MDR, are they totally sensitive to all drugs?
They are not. They're just not MDR.
Oh, but they could be resistant to some drugs.
It could be resistant to some drugs. So I'm going to ask, okay, so one last question for Manny, and then I'm going to ask that we hold questions, because this is still the first slide.
Go ahead.
So what I'm illustrating here are two normalized histograms. On the left-hand side, we're comparing clinical isolates that were multi-drug resistant, which are defined by resistance to two of the first-line drugs for TB. These are isoniazid and rifampicin. And in black, on the left-hand plot, are all the other strains that are not multi-drug resistant by this definition. And that means they can be resistant to just one of those two drugs, or they can be resistant to lots of other things, but they are not multi-drug resistant. Because multi-drug resistance bacteria, TB, is defined as resistance to both drugs, isoniazid and rifampicin, and because isoniazid, so it's defined that way. And what we further did is we looked inside that large data set, again, commissioned by the WHO, at strains that were only that possessed resistance to isoniazid, or that possessed resistance to rifampicin, the other arm of the multi-drug resistant. And what we saw was just that this phenotype was preserved in just the strains that had some kind of, at least had isoniazid resistance. And what's important to know about isoniazid is that as a drug, its formulation is actually a prodrug that becomes is inactive when it first gets taken up by a cell. And for that drug to be activated, there's this enzyme catalase, which is really a peroxidase that will cleave, will cleave part of the isoniazid prodrug, add this NAD adduct, and that becomes the active form of the drug. And that drug then will bind proteins that are involved in the synthesis of the TB cell wall and cause a cell to die. And so something like 70% of mutations that cause isoniazid resistance occur specifically at catalase, specifically at a single site, which you'll see later called S315T, so the serine at 350 position to 3. And like, another whatever 15-20% is in other spots on isoniazid, and then if it's or on catalase, and it's not on catalase, it's in the drug product. So what I'm trying to just summarize here is that isoniazid resistant clinical isolates replicate the phenotype that we see in multi-drug resistant clinical isolates, in part because multi-drug resistant clinical isolates will also be isoniazid resistant. And the main clinical mutation associated with this is a mutation on catalase. And so to kind of validate these meta-analyses that we've seen, what Nana had done was we took some clinical isolates that were accessible here in the PHRI from a different collection of TB clinical isolates. And what we did was we performed assays where we took TB cells that were growing just kind of healthily in log phase and treated that were either multi-drug resistant or not multi-drug resistant, or drug resistant, not drug resistant. And we treated them with isoniazid and looked to see how much survival there was after 30 days. And so what you see on the left are measurements of surviving TB cells that are capable of forming colonies, either in black, cells that were not multi-drug resistant versus cells that were orange, cells that were, am I right? Yeah, so in black and gray, these are cells that are susceptible to isoniazid. Right, so these are not isoniazid resistant, whereas in orange and red, you see clinical isolates that are resistant to isoniazid. And what we see here is that there is more killing in the orange and red strains in general, right? So this validates that isoniazid resistant clinical strains are more susceptible to bedaquiline than the isoniazid susceptible ones. Furthermore, I want to highlight, because it's something you're going to see in the rest of the slides, that the orange strains are multi-drug resistant, and the red strain is going to be a clinical strain that is mono-resistant to isoniazid. So because the key, most important mechanism for isoniazid resistance is a mutation, a loss of function mutation, or at least a partial loss of function mutation in catalase. This led us to a series of hypotheses that really was this deficiency in the catalase enzyme that was causing the phenotypes that we saw here. And so the rest of the story is really about that mechanism. And so the first thing that we did is we from a colleague, performed experiments in a clean CatG. So CatG is a gene that causes expression of catalase, whereas that is catalase. And what you see here are dose response experiments. Black is our control, and in general, orange is going to be our isoniazid resistant strain, right? So what you see here is that our isoniazid resistant catalase deficient cell are hypersensitive to bedaquiline, because you see this leftward shift in that dose response. And importantly, this is something that can be complemented. So if we we express CatG back in those knockout cells on plasma constitutively, that we get this recovery and actually this overshoot of protection from bedaquiline. So that's the rightward shift in that dose response. On the right-hand side is kind of validation that indeed these strains are either isoniazid susceptible or resistant, right? So the black strains are susceptible whereas the orange strains are resistant. So none of these concentrations that we tested were able to kill, these TB cells. And if we complement, we get a partial recovery. As further kind of validation and to test the hypothesis, so with the collaborator, Shi Ma over at the University at Seattle Children's Research Institute, what we did was we performed experiments where we overexpressed this transcriptional regulator called FIR, regulator. And what's special about FIR is that it's an inhibitor of the catalase chain of CAT-G. So what you see is that we see very similar phenotypes whereby cells that have FIR induced, right, are ones that are going to be decreased in expression of CAT-G, therefore decreased in expression of catalase. Those guys are protected. Those guys are hypersusceptible to bedaquiline, right? It's going to be qualitatively similar to deleting or mutating in the CAT-G gene. You can see that because the orange bar, orange line is lower either in the 30-day survival curve on the left, which we saw with our CAT-G deletion strain, and then on the right where their lab had overexpressed this FIR gene. That partially validates our hypothesis. So now we became interested in understanding mechanism. So some of the other important things to know about catalase is that its primary function is to really to detoxify reactive oxygen species, more specifically peroxide. And so we hypothesized that, well, if this is a primary activity catalase, then likely getting rid of that activity would make these cells in general hypersusceptible to loss. And maybe that may be related to why these cells are also hypersusceptible to bedaquiline. And so what you're seeing here are some experiments that we did. So for the next several slides, the plots on the left are going to be with lab strains, whereby our drug susceptible or ionized susceptible strain is the wild type laboratory strain, H37RD black. And in orange is going to be that CAT-G deletion strain that I was talking about. And on the right-hand side are these TB clinical isolates that I showed you a few slides back that we worked in here in the lab. So left is lab model, right-hand side is like this kind of validation of clinical strains. And so the first thing that we did was to test that hypothesis. We'll perform these peroxide dose response growth inhibition experiments. And what you see in both of these cases, the drug-resistant, isoniazid-resistant strains, including the multi-drug resistant clinical strains, are hypersusceptible to peroxide than the black isoniazid susceptible strains. You can see this by that leftward shift, the dose response curves. And again, in particular, I'm going to call attention to this red monoresistant strain, which can function a little bit as a control in interpreting these results.
But that should not be specific of isoniazid resistant strains. I mean, any blood type or TB will be more susceptible to hydrogen peroxide if catalase is affected. It shouldn't be a specific, right?
Catalase, the way the biochemistry for catalase works is that it is specific to peroxide.
No, yeah, of course. What I'm saying is that this phenotype is likely not specific to isoniazid resistant or drug resistant activity.
Yeah, so let's come back to that afterwards, because I really don't, Marcelo, we can talk about that afterwards. And I'm really gonna probably not gonna stop for questions, so I can get further in. So we became interested. So we have evidence that says that these isoniazid susceptible strains, whether in the lab model or these clinical strains that we tested, hypersusceptible to also hypersusceptible to ROS, right? And because catalase main activity is to detoxify ROS, to get rid of ROS, to get rid of ROS, right? We expected that, well, there'd be more evidence of increased oxidative damage to the cellular components. And so what we did was Mustafa Shaikh in the lab, an undergrad in the lab at the time, performed these ELISA experiments to look at oxidized. I'm sorry, this is not the right slide. That's the next slide. So I guess I'll take a step back and say that in addition to that peroxide validation assay I showed you before, previously we also did some RNA sequencing, and so kind of consistent with that expectation that you'd have if cells are deficient in their ability to detoxify ROS. And what you would expect is that ROS sensitive, oxidative stress sensitive genes would be upregulated. And indeed, that's what we saw. So on the left-hand side, in drug susceptible, so this is the wild type cell, that the presence of bedaquiline induce expression of CATG. So it turns on the oxidative stress response regulator that increased expression of catalase, this ROS detoxifying enzyme. And that if we used a BSL-2 safe strain that similarly has these phenotypes, where the black is isoniazid susceptible and the orange is isoniazid resistant, that also phenocopies the hypersusceptibility to bedaquiline, that we can perform assays kind of in our BSL-2 space to measure changes in the accumulation of ROS, which we did here using this kit CellROX that fluorescently labels accumulation of ROS. And that, if we perform ELISA experiments, you can also measure the increased abundance of proteins and DNA that receive oxidative damage. And so what I'm showing you here is that the gray bars are our drug susceptible strains, clinical, laboratory clinical. And each pair of bars, you can see that the bedaquiline treated cells have increased accumulation and damage than the wild type, than the untreated cells. So this led us to further explore mechanisms for why, based off of like our RNA sequencing data, we began kind of exploring other potential mechanisms by which this catalase deficiency might be causing the bedaquiline hypersusceptibility. So some of the things that we noticed was that there were several transcriptional programs that were upregulated. And so one of the things that led us to hypothesize that perhaps what's happening is that as kind of a stress response to seeing this new drug, right? That TB, just like every bacteria, will turn on different sets of transcriptional programs. And then maybe some of these transcriptional programs themselves, through whatever kind of set of genes that are in their regulons, that the induction of some of these transcriptional programs might also be causing these cells to be hypersusceptible to bedaquiline. And what we did is we looked at kind of the top three most abundantly overexpressed transcription factors. Our colleagues, over in Seattle, tested these three strains, tested strains that are overexpressing these three transcription factors. And what we found is that, for two of the three, we saw bedaquiline hypersusceptibility, and this is the one that has a stronger phenotype. And so both at the level of, in the case where we're measuring the amount of ATP that is present inside the cell, which again is downstream of the target bedaquiline ATP synthase, as well as the ability for those cells to grow. These strains that have these transcriptional factors programs turned on are hypersusceptible to bedaquiline. So
What
again,
transcription factors? Can you define please?
yeah, a transcription factor is a protein that
No, that I know. Which one?
this one 3160C. Okay, so I'm going to really be more vigilant about not taking questions. And so, to further validate this, we kind of looked in the literature about this transcription factor. Very little is actually known about RB3160, but it turns out that one of the proteins, one of the genes in its regulon, 3161C, has been previously reported to be associated with differences in isoniazid susceptibility, which again, like our phenotype for bedaquiline is between the isoniazid susceptible versus resistant. So these are qualitatively suggestive that this transcriptional program, at least by association through this drug isoniazid, may explain some of our phenotype of bedaquiline. I told you a few slides earlier that these isoniazid resistant cells are hypersusceptible to peroxide, to ROS. In addition, what I showed you is that when we performed ELISAs, we saw evidence of increased accumulation of oxidatively damaged DNA. And so this, in the presence, following treatment with this drug bedaquiline. So this led us to also hypothesize that perhaps these cells are succumbing because now they're accumulating more DNA damage, right? And so, the evidence for this is supported by, these changes or this induction of DNA repair genes in the isoniazid resistant orange strains following bedaquiline treatment. Right, so these guys are sensing DNA damage and turning on expression of the DNA repair enzymes. So you see on the left. And to test our hypothesis, what we did was we took these strains and we treated them with a DNA damaging agent, performed dose response growth inhibition experiments. And that's what you see on the right, that these isoniazid resistant, multi-drug resistant, and singly drug resistant clinical strains also are hypersusceptible compared, orange and red strains are hypersusceptible to DNA damage than the isoniazid susceptible strains in black. I think for the purpose of simplicity, because kind of getting wordy, I'm just going to say black versus orange and red. Hopefully that'll make things a little bit easier. Okay, so some of the other things that we noticed was that there were a lot of gene expression changes in different aspects of metabolism. And as systems biologists, one of the tools that we have at our disposal are modeling tools that allow us to predict what might be happening to metabolism in the cell, given different observations that you have, including changes in gene expression. And so, without going into a tremendous amount of detail, what we did was we performed metabolic simulations using some of these genome scale models of metabolism. So we're talking about models that encompass about 1,000 different genes inside TB. To predict how changes in activity of different reactions may be, how the activity of different metabolic pathways and reactions may be changing following treatment with bedaquiline. And what I probably should have stated earlier, but what might have been obvious from previous slides, is that again, we have our wild type in gray and catchy deletion strains in orange. And we have untreated and bedaquiline treated samples. And so what you see here are simulated or predicted results for the activities through different metabolic reactions that are part of folate biosynthesis. And for those of you guys who may not remember biochemistry, folate biosynthesis is the metabolic pathway that provides potential hydrofolates, which are early substrates for the synthesis of nucleotides, bacterians, protein biosynthesis. So, a lot of anti-cancer drugs target folate metabolism because if you shut down the ability of a cell to be able to synthesize nucleotides, then they're really good at either preventing a cell from growing or killing cells. But in any case, what we saw here, you can hopefully you can perceive this, is that our orange cells following bedaquiline has significantly less activity through these folate biosynthesis reactions than our our isoniazid susceptible gray cells. And so to validate this, again, we perform these experiments in clinical strains. So there are these two drugs, trimethoprim, and I always have a hard time pronouncing sulfamexazole, SMX, that inhibit two different steps in the folate biosynthesis pathway. And what you can see that was interesting is that in the case of the red strain, so these are clinical strains, the clinical strain that is only resistant against isoniazid, that that at least similar modeling pans out that this strain is hypersensitive, hypersusceptible to folate biosynthesis inhibitors, right? And just a reminder that our orange strain here was RNA sequencing data from just a catalase deletion strain. So it didn't have a mutation that would cause rifampicin resistance. But here in orange, which are multi-drug resistant strain, clinical strains, these guys didn't have a change in phenotype, in the case that, in response to folate biosynthesis inhibitors. So this is an example of how this physiology changing in TB caused by the acquisition of resistance to this drug isoniazid, so half of the definition of multi-drug resistant phenotype, and that it doesn't have changes in physiology, some change in physiology associated with resistance to other drug, and that in the preceding slides, the changes in physiology that were associated with resistance to that first drug can explain mechanistically why multi-drug resistant TB is, susceptible to this new drug, bedaquiline, but not to these folibastinus inhibitors. And so to summarize, I know there's a lot of jumping back and forth in terms of names of drugs, but what we've shown is that multi-drug resistant TB, clinical isolates, are hypersusceptible to this new drug, bedaquiline, ATP synthase inhibitor. It's a cornerstone now of how we globally treat multi-drug resistant. And some of the reasons why that is because they have a hard time detoxifying ROS, have a harder time dealing with DNA damage, and they also turn on some transcriptional programs that also may be sensitizing them to these new drugs, through mechanisms that we don't fully understand yet. And also that that in the case of the multi-drug resistant cells, why they don't share that phenotype that we have seen with hypersusceptibility to folate biosynthesis inhibition, is that acquisition and that second set of mutations that cause resistance to ripepsin is somehow compensating for that hypersusceptibility to folate biosynthesis inhibitors, again through mechanisms that we don't understand, but which is really interesting. So now what I'm going to do is I'm going to switch gears a little bit. So that last part, that first story was a story really about metabolism. And what I'm going to do in the second story is tell you a little bit about more about things that we've learned, tools that we've been developed to understand transcriptional stress responses inside TB. And this is a project that in my lab was led by Oliver Gu and Edson Petrie. Oliver is a part-time medical student here at New Jersey Medical School. And Edson is an undergrad who's now at Rutgers, New Brunswick. And this is a project that was in collaboration with our collaboration Yuma and postdoc in her lab, Ethan, and a grad student, Brady. And so, again, I'm just going to give a little bit of a primer, help us understand bacterial physiology. So I told you at the beginning of this talk that stress responses in bacteria are really organized just in these things that are called regulons. And most of these regulons immediately follow the promoter for a transcription factor. Although for a general stress response, transcription factor, they may have regulons kind of sprinkled throughout the chromosome for bacteria. Some of these general stress response, transcription factors, right, are things like sigma factors that help another transcription factor dock. And so those more clearly would be understandable why they would be distributed. So you have in bacteria, you have these things that are called regulons, right? And they get turned on by a transcription factor. Some of these regulons contain genes that are also transcription factors that control their own regulons. And so now when you start to cascade, and kind of, if you were to on paper try to map out, how information may flow, transcriptionally through a cell in response to some set of extracellular cues, these kind of structures become complicated because a lot of these regulons, crosstalk or interact with each other, and eventually form, these very, very large and dense networks. And so a consequence of having gene regulation, eukaryotic cells are the same thing, right? A consequence of having gene regulation, be structured in the form of these really dense, complicated networks is that it then makes it really, really challenging to understand, what specific transcriptional program or what specific, transcriptional mechanism may be causing a phenotype that you care about, especially when you're doing things like looking at RNA sequencing data. And a lot of times when we do bioinformatic analyses, right, we just, we look at, the most differentially expressed genes, and we may do some sort of geontology analysis and hope that if we see transcription factor in that, twofold change and low p-value, then we may say that we hope that transcription factor is being induced. But not all transcriptional programs require that, not all transcriptional programs require that a transcription factor have really high abundance, right? So the promoter, induction of a regulon is really driven by the activity of a transcription factor, and not all reactions are driven by the abundance an enzyme as an example, but can be driven by this kind of turnover, right? Difference in turnover. And so what we decided to do was to create some computational tools to help us be able to analyze change in the activity of different transcriptional programs in TB given RNA sequencing data. And our overall strategy was to create what's called a gene regulatory network, which is literally a map of this network of all the transcription factors and all the genes that they regulate, and to use such a gene regulatory network to be able to decipher what sets of transcription factors may be highly or lowly active for any gene expression profile that we saw. And so our starting point was that, again, our collaborator has this library of strains where every member of that strain, of that library, is a strain where there's one transcription factor which can be inducively overexpressed. And what our colleagues had done was, kind of in a previous publication, when they synthesized this library, it was like a long time ago now, they had performed microarray studies to look at changes in the global gene expression of TB following induction of any one of these transcription factors, right? So what happens in these microarray data sets is that you turn on one transcription factor, but it's not only the genes in that regulon that change expression. You get lots of different changes in expression across the cell. And again, that's because some regulons have genes, right? And some of those genes will regulate the genes of some regulons are transcription factors. And changing the abundance of those transcription factors will change the abundance of other genes, some of which are transcription factors in that cascade. And so what we did was we first started with experimental data from those experiments. And on the right-hand side, what I'm showing you is descriptively just a sense of just how diverse this data set is. Right? So TB, roughly 4,000 genes, roughly 200 of these are transcription factors. We have about 200 different strains here at different points. And what we're looking here on the right-hand side is kind of like a UMAP. Well, it is a UMAP, but it's kind of like a clustering visualization of how many different configurations of gene expression are there in this specific library. And so what you can see is that there are a lot, but there's also not that much, right? So there's only maybe about a dozen different clusters that you see here. Now, for the purpose of being able to generate a gene regulatory network computationally, what's required is a large data set where there's lots of observations of gene expression, a large data set that has to be very biologically diverse. Because if you have because the computer won't be able to see won't be able to sense an interaction between a transcription factor and a gene if, for instance, that transcription factor is never induced or that gene is never induced. So you have to have a biologically diverse starting point. And so even though this library, this data set might be kind of what you expect as the gold standard because, hey, we're turning on each transcription factor, you can find a signature for each transcription factor. Turns out that the signatures are really not that diverse. Some of these genes are almost never going to be turning on. And so to deal with that, what we decided to do was we decided to do some analyses where we took all of the publicly available RNA sequencing data sets that are available on Sequence Read Archive at the time. So this is, again, roughly 4,000 different RNA sequencing data sets. And these are collected by labs all over the world, all sorts of different treatment conditions, following different clinical strains, lab strains, drug treatments, growth environments, salt, hypoxia, et cetera, different kinds of environments. And what you can see here from the UMAP projection on the right is that this data set is incredibly biologically diverse. And so each one of these clusters here is kind of a different configuration of gene expression of TB in whatever that set of samples are. And some of these samples can come from across different labs, across different studies. So what you might expect probably in one of these big orange, green or red or brown clusters are going to be, all of those control conditions that everybody uses, which is the same, wild type cell and exponential phase and the same media growing, aerobically. So then we did a little bit more descriptive analyses. We looked at, on the left-hand side, how much if we were to combine, our microarray data and our, this very large component that got from RNA sequencing, including some important normalizations so that we can make these data sets agree with each other, where you can get a sense of how much more biologically diverse our new compendium is based off of RDC. gray versus the red, which is just the microdata. And on the right is some descriptive statistics on how much variation, like how much change there is for any one of these 4,000 genes. So on the x-axis is the median level of expression across all 4,000 samples. So what you see is that some of these genes are going to be very highly expressed at all times. So that's going to be an example, would be this top A gene on the far right. And some of these genes are going to be across the whole compendium, almost never getting turned on, right? That's something like the GAL-K gene that you see on the left. On the y-axis is they measure the variance inside our data set. So there's a median, or I guess it's the mean absolute deviation. So this is basically, it's kind of like if you think about like statistics, we measure like the mean and standard deviation. The median on the x-axis is very similar to our mean. We have a large distribution that's not a normal distribution. And the absolute deviation on the y-axis is very similar to the standard deviation that you would be calculating for a normal distribution. And again, you see that some genes are very, very highly variable, right? Like this HSP X, right? And many of the genes are not, right? So genes that are more highly variable would, you would intuit that these are genes that are part of stress responses, right? Because usually if there's no stress, those genes don't have to turn on. But if there is a stress, then they have to turn on, right? So, but they're not going to turn on all the time. So, so we did some descriptive analysis to kind of look to see what's here. And it turns out that in general, transcription factors like in TB have, have a larger MAD, meaning they're more variable. That's kind of consistent with the expectation that they're only turning on, or only get induced when they see certain kinds of environmental stressors. So, so what we did was, again, like what we were trying to do was assemble this gene regulatory network so we can use it kind of like to decipher, what TB is doing under different conditions. So it turns out that there's a suite of tools that you can use to estimate gene regulatory networks. These are kind of different kinds of machine learning analyses or multivariate statistical analyses. And long-winded way here on this slide of saying is that what's been shown is that use any one method, no single method is perfect, right? Because the different methods have different math. There's different kinds of assumptions that are used in each different method. But it turns out if you were to take lots of different methods and look kind of, in an ensembling approach, look to see what kinds of connections are common between different methods, then you can have more confidence that whatever gene regulatory network you get from the ensemble is likely to be better than what you might get from any of the individual methods. Right, so we took this ensembling approach that was published in Benchmark, I guess now 10 years ago, and without going into a lot of detail here, what we constructed was a network that had about 60,000 direct and indirect interactions. So direct would be that operon, right, that regulon, and indirect would be transcription factor turns on somewhere down the line through other transcription factors that's changing the activity of those genes. And what we could do then is use this new gene regulatory network to, just make sure this is the right slide. Yeah, so what we could do with this gene regulatory network is basically deconvolve or decipher the RNA expression kind of data sets that we have. So what I'm showing you here is when we apply that to the that microarray data set that we started with, what we see is that, well, there are some transcriptional programs, So when we do this analysis, what we get is transcription factor activities. So I probably should have clarified that. We get this large gene regulatory network, and we take our gene expression data set and we divide it by that network. So, so we start with, say, as an example, let's say there are 1,000 samples and there's 4,000 genes in TB. Our gene regulatory network is 200 transcription factors by 4,000 genes. And so we do something that looks like a matrix division. And for each of your 1,000 samples, you get activity levels for the 200 different transcription factors. So we're going to call that a TFA. We're going to call that a transcription factor activity. So we do this kind of analysis of a microdata set, and that's depicted here. So these are, again, the median MAD, so like kind of like the mean standard deviation of each transcription factor activity across the microdata set. Now, if gene regulation was very simple in a cell, what you would expect is things to be kind of a diagonal, right? The more expression you get of a transcription factor, the more activity that you would get, right? That's not actually the case. That's not always the case because some of those transcriptional programs are highly constitutive, right? That are maybe not sensing something in the environment and just always kind of like housekeeping transcription factors. And so what I'm depicting here are kind of the median and the MADs of like the transcription factor activities. And what you see here is that, actually, I'm sorry, this is the whole RNA sequencing data, not just the microarrays. Is that there are some transcription factors that have very low MAD on the bottom, or excuse me, on the bottom, low across the y-axis. These are transcription programs that are likely to be more constitutive. And then you have, if you were to look on the x-axis, or on the y-axis, ones that are very high in MAD. These are going to be transcription programs that are highly responsive to stress. And we annotated some of those, and indeed, that's what many of these transcription factors are. And just example, SIG-A is like a sigma factor. So again, this is something that allows transcription factors. This is an inducible, sigma factor that can recruit a bunch of different transcription factors or promoters. DEV-R is a very, very well-studied stress responsive transcription factor. So this is just an illustration that the amount of diversity in the transcriptional activities of different transcription in the cell is much less than the total diversity of gene expression changes there may be. So again, these analyses are across all 4,000 publicly available RNA sequencing data sets. We're just looking at UMAP projections of just the RNA profiles alone versus transcription factor activities alone. Yeah, so I'll kind of, in just time, I'll just go a little bit further. So, so now we have this tool, right? We have this gene regulatory network tool that we can use to estimate how active any one of these 200 different transcription factors are if you give me any RNA expression profile. And as a way of using this tool, what we wanted to do is say, if I have this tool, can I use this tool to help us understand molecular mechanisms that explain TB's response to stress? And ultimately, I want to study drug stress, but in this case, I just want to ask generally for stress. And so what we did was we applied our tool to the transcription factor overexpression library, the microarray data set. And actually, I guess what I'm showing you here is a correlation between expression. So we applied this tool to the transcription factor microarray data set. So now for each of the different 200 strains, we have estimates of 200 transcription factor activity. What we also have depicted in the cartoon are measurements of each of those 200 different strains if you pull them together, in just normal growth media. And so what that experiment gives us is that for each of these 200 different strains, where one transcription factor was overexpressed or highly induced, right? How well does that cell grow? So if you turn on some transcription factor programs, what it might do is shut down metabolism in the cell. And what shuts that down, that cell is not going to grow very well. It's going to just go to sleep. And there might be other strains, other transcription factors where you turn that on. It can take up a lot of, nutrients and grow really fast relative to, most of the other strains, right? So what we did was we used those growth phenotype measurements of each of these 200 different strains. So for each of these 200 strains, we have something kind of like a fitness score, kind of like a growth rate, maybe you can call it. You can think of. And we looked at these 200 different transcription factors and built this random forest machine learning model to be able to predict how well will that TB cell grow if it has whatever combination of transcription factors that are active. So on this side, what I'm showing is that we built a machine learning model using that gene regulatory network and using these measurements of these phenotypic measurements of what transcription factors do to cell growth, the cell thickness. And on the right-hand side is showing you that when we train the model, what does that trained model do in predicting the data that we're giving? Really, like, it should do very well. And just kind of as a statistical validation, does do very well. So now what we want to do is use this tool, use the machine learning model. What we want to do is validate this machine learning model, right? So we can do the statistical computation analysis. We can get something out of this, but is a model actually able to, predict phenotypes that doesn't actually that weren't directly given to the machine learning model when it was being trained, right? Can this thing make predictions based off of information it's never seen before? And so to validate this model, what we did is we took public RNA sequencing data from TB cells that were grown kind of happily, aerobically in kind of nutrient-rich conditions, and that then were subjected to hypoxia, right? So when you take away oxygen from the environment, the cell's going to make ATP much worse. I guess that they're not able to make ATP, but they make ATP much worse. That is going to turn on. That does turn on a bunch of stress responses, which collectively, causes Mycobacterium tuberculosis to become dormant and just kind of hide out, right? And then in this experiment, what happened was, the oxygen was reintroduced into the environment as reaeration. And on the left-hand side are measurements of the density of colony-forming units of TB during the hypoxic phase and in the reaeration phase. And so in this left portion where things are kind of flat, what you can see is that hypoxia causes kind of a growth arrest, or maybe some cells are dying. I guess there's kind of an overlap with the standard deviations, but basically, hypoxia will arrest the growth of those cells, and then once you reintroduce oxygen, at this point here, the cells will be able to about halfway through at day 7, the cells will be able to rebound and start growing again. So we have RNA expression profiles for different days from that experiment. And what we then did was we used our gene regulatory network to estimate what are the activities of these 200 different transcription factors at each time point where we have an RNA sequencing data set. And then use our random forest machine learning model to predict how well is TB growing at that time point when that sample was collected. And what you see on the right-hand side is a prediction by that machine learning model showing that during the hypoxic phase, over time, the fitness or the ability of those cells to grow is decreasing. And after oxygen is reintroduced at day 7, it starts to rebound. I don't want to overinterpret this because obviously this is not perfectly matching what you see on the left-hand side. But there's at least some qualitative validation this analysis that this machine learning model can predict these phenotypic changes in the ability of these cells to grow, just based off of the transcription factor activities alone, and just based off the RNA sequencing profiles alone. Now, what's really cool about this approach is now we have this model, right? We have this machine learning trained model that is comprised of transcription factors. And so if this model is good, right, then what you would expect is that transcription factors that the model says are really, really predictive of whether or not a cell is going to grow, in this case, under hypoxia, they should be mechanistically related or involved in controlling TB growth under that environment, right? The null hypothesis of that would be just that you get spurious transcription factors defining defining that model. And so what we did was we went inside that random forest model and looked to see which transcription factors are the ones that are really that the computer is using to make these decisions about how well will TB grow. So for each of these 200 transcription factors, we have some sort of numerical coefficient. They have some directionality. And what I'm showing you here on the right-hand side are the top 7 different, top 7 different ranked transcription factors that the model says can predict growth under hypoxia by magnitude. 7 strongest transcription factors by magnitude. And what you can see is that for each of those top 7, there's literature support. And this is something that, again, the computer has never seen. The computer doesn't know literature. It doesn't actually know what hypoxia means. It doesn't even know what fitness means. But what I'm illustrating here is that we can use these approaches to actually pretty directly get to experimentally testable hypotheses, which in this case, all these other papers in the literature tested, right? validating that this approach can, validating that we can get molecular mechanisms from this approach. So I'm going to summarize this by really just saying that, you know, on this latter part, even though we have not yet studied drug responses, that's kind of like the next step. What we've done is that we've at least built this tool that allows us to, number one, estimate transcription factor activities for any RNA sequencing profile. Number two, allow us to train machine learning models to predict, you know, how well, whatever phenotype could be drug susceptibility, could be growth in hypoxia, could be something else, given the RNA expression profile. And then three, we can actually use that machine learning model to help us kind of, you know, generate hypotheses that can be experimentally validated. And then I guess I'll just kind of end there. I think the people would like to work on the work. So that first story on metabolism and drug resistant TB, those are the names in red. That was really led by Nada Floriani in the lab, who's now postdoc in Pemi Salgame's lab and other collaborators. A second story on the right are the folks labeled in blue. So, yeah, I thank you for your attention. So we are just past 1 o'clock. So I won't be offended if you guys have something urgent, but happy to take any sets of questions now. So David and then Manny.
Two questions. So catalase modifies isoniazid.
Yes.
So it shows that it doesn't have the exclusive specificity for hydrogen peroxide. So do you know that catalase does not inactivate the dactylen?
Okay, so the statement was that catalase acts on isoniazid, so therefore it may not be fully specific, may not be exclusively specific to peroxide. And the second is that in light of that, is there evidence that it is not directly active on the dactylen? So I guess that first statement, I guess that's a true statement. Although I would say no enzyme is only specific to any one molecule, right? So yes, varying differences in like affinity, right? But I forget the chemistry, but yeah, I guess yeah. In this case, like catalase uses, I believe, NADH as a cofactor. So I guess it does, but it with much less specificity than peroxide, or much less affinity than peroxide. In the case of this catalase,
I guess my question really is, do you know that it's not acting on the dactylen to inactivate it, and therefore a catalase deficient strain is going to be more susceptible to being acute?
So on the question of like, is catalase causing dactylen sensitivity by not, but is a catalase deficient cell causing dactylen sensitivity by not detoxifying some of the dactylen? We don't actually have an answer for that. So, and I don't really want to speculate anymore. Yeah.
And the second question is with the hypoxia study. So is this specific for hypoxia, or is it just growing in non-growing cells?
That is specific for hypoxia. So the question is like the hypoxia study, some questions about the experimental design. And so the experiment begins with exponentially growing TB cells that suddenly experience a change into hypoxia with fresh media and kind of like the same, nutrient condition. So these are not cells that begin in stationary phase, and they're not cells that are nutrient deprived.
But if you could make TB stop growing in some other way, right? Gradually stop growing, with the same set of transcription factors change their activity.
So the question is, in a different model where there was another mechanism used to slow down TB growth, are those same transcription factors predictive? Because I guess it's whether or not there's any specificity for, are those transcription factors sensing anything more specific to hypoxia than anything else? I would say the answer is no. Some of these that we know are, sensitive to, lots of different things. So KSTR is an example. MSE2R, these are sensitive to metals. So they're also going to be sensitive to oxidative stress, metal stress. But I don't know enough about every single one of these to be able to say. I mean, I think that given how we understand bacterial physiology, some transcription factors will be highly specific to a limited number of stresses, and some are going to be more. So, and it's, you know, when you have different forms of, you know, growth restriction, you know, the stresses are different. The stationary phase, like, you know, is not necessarily the same as head loss. So Manny, next.
So getting back to the first half of your talk where the catalase increased the sensitivity to the drug, can you get that slide up? It was one of the early slides, or even on the summary slide of the first half. Yeah, right. Right. So this may be a stupid question, but I don't understand why the lower concentrations of the BDQ give you more inhibition, and then it goes up with when you have more growth, and then you have inhibition again. Why is it a bell-shaped curve? Why isn't it complete lack of inhibition at the lowest concentrations of BDQ?
Yeah, so the question is, I guess kind of a related question is, why is there overlap in the curves? And why is, you know, why are these two bell-shaped curves? And why is it not something that might be all of the MDR strains are very, very susceptible to that one?
Yeah, I mean, the points on the left part of it, closest to the y-axis are low. And it should be high, and then it should go down as the concentration increases.
I think there's a misunderstanding of the way the plot is drawn.
Am I
It
wrong?
actually is a histogram and the bars are hidden. And the maximum effect is where the peaks are highest, right?
So,
It's the normalized number of strains at each point
Not
so,
actual concentrations, the number of strains that are more
on the y-axis.
sensitive or persistent.
right, so Manny is asking, why are most of the orange like strains not more skewed to the left than in this plot? And so to explain that, this data is every strain has a measurement of the MIC50 of like the minimum inhibitory concentration. That means as you go to you know higher concentrations, right, they will still be growth inhibited. And at lower concentrations, is that right? So lower is to the left, then they'll be less growth inhibited. So for each experiment, right, there is each of these data points is basically a dose response. And each of these has a numerical value. And so this is a histogram of the value. Why are there not more strains that have a unique MIC at a lower concentration? So as you go to the left, that is when the concentration of bedaquiline is lower, right? So that means that more of those cells are going to be able to grow fine. And as you go to the right, is that right? No, I'm sorry. Yeah, you can tell like my blood sugar is really, really low, so forgive me for that. So a leftward shift, right? This is kind of this is a dose response, so shift to the left, right? So at the very, very low concentration,
No inhibition.
no. So basically, each of these points here is a number of strains that are inhibited at that concentration. So at the lowest concentration, all the way to the left, there are very, very few strains that are actually inhibited at that concentration. So that means in either case, there's very, very few counts of strains that are inhibited at 8 nanograms per mil. And so what's not shown here is a cumulative. So as you keep going higher, what you would expect is actually, instead of having this be a bell shape, you would have something that kind of plateaus. This is not a cumulative distribution. This is just a distribution of just the individual MIC values.
So each point incorporates a single MIC from a single strain, rather than following one strain across different concentrations.
No, it's a mixture.
No.
It's 150.
Each point is many
Many
strains.
strains.
Well, no, at each point, but each individual number comes from a single strain, and they're averaged, right? And each
Each point comes from is the number of
So-
strains that have that MIC
right, so
normalized to the peak.
a single data point for each strain.
So
We
this
can
is
go
say
back to
something
the paper, right? It comes from the paper, right?
came from like a large. Yeah, okay. So on the left-hand side, most 10% of the strains, no matter what you're looking at, cannot be growth inhibited by Biochemistry. That might be a better interpretation. No matter what 10%, 10% of all the strains are not inhibited by growth by Biochemistry.
Jason, I have a question about, sorry, you had your hands first, but what I want to quickly ask is about this machine learning approach. Your original idea of those 20-some strains that for which microarray data was available is interesting, and you could create a model based on that. But then you added 4,000 other strains and RNA-seq that presumably didn't have any transcription factors upregulated in them. What is the rationale for including all those in the model?
So I just want to make sure I understand the question. So the question is, I have number one, a data set, 200 transcription factor strains, the 200 strains for transcription factors are induced.
Yeah.
Right, and I can do my approach and I can calculate the activity of transcription factors, right, for each of those 200 strains. Right, so your question is, why do I need the extra 4,000? Why do I need the RNA-seq compendium with the other 4,000 samples? Or
You can explore what happens in those, but they're different. Nothing was induced in those, and they might have been subjected to some other condition. You got it from various literature, right? And they were RNA-seq. So there are two different kinds of data that you're clubbing together. And you expect it to do some realistic predictions.
So the question is, in a larger set of 4,000 samples, right? In those experiments, transcription factors are not singly induced, and we don't even know if those transcription factors are induced, and therefore, would it be reasonable to expect that nothing is changing or nothing is different in that part? So the answer is no. Because when we look
You're
at
training on them, right?
well, I think what I want to I think maybe this was my part very unclear, is that across our compendium, right, there are transcription factors. This on the right hand side is a depiction of expression, the mean expression, the MAD of each transcription factor across the 4,000 samples. So some transcription factors are highly induced on the right, some are lowly induced on the left. Some change a lot at the top and some change very little on the bottom. So transcription factor activities do change across the compendium, and that's what would be expected because these 4,000 samples are subject to very different experimental conditions. And so what we did was, in the first part, we built a tool. Our machine learning model is a tool that says, hey, if you have whatever configuration of 200 transcription factors, what is the likely growth state of that cell, or the ability of that cell to like grow or respond?
And you know the likely growth state of these 4,000 samples?
So the question is like, do we already know the growth state of those 4,000 samples? So the answer is no, and I want to clarify maybe where I may be causing some confusion. So data set number one with the microarrays and data set number one with the growth experiments, those are data used to instruct a model, instruct a machine learning model. I'm proposing that we can use that machine learning model to predict the ability of TB to grow in any of the other 4,000 samples.
That's what was the confusion. So you have a training model and you want to predict on something else.
That's
So
right.
those 4,000 were targets of prediction.
Those 4,000 are use case. And the hypoxia is an example of a
Just
use
yet another
case.
example.
Yeah, is a
Then I
validation use case.
understand. So I just looked at you.
No, that's my fault. I forgive the miscommunication. Marcella and then.
I have two questions. So are catalase deficient strains hypersensitive to other antibiotics besides beta-alanine?
Yeah, so the question is like, are catalase deficient strains hypersensitive to other antibiotics? The answer is yes. Because I showed 3 of them. Fluomycin, trimethyl, sulfamycin, Bacillus, yeah. So the answer is yes.
So why is your focus on beta-alanine?
The question is why focus on that? Well, that's because that's where I started. That's where we started, right? Descriptively, we saw that in this collection of multi-drug of 12,000 clinical strains, we saw a phenotype of multi-drug versus not multi-drug. And catalase deficiency is 1/2 of the, the definition of multi-drug. And we didn't have this phenotype with the other half, the rifaximin resistance.
Yeah.
So that's why we started with that.
Yeah, the second question is, a portion of isoniazid resistant strains have mutations in alkaline hydroperoxidase, OA.
So there,
Are those also more hypersensitive to antibiotics?
so the question is, there are other forms of isoniazid resistance, which include mutations in another prostase, AHP. Do those have the same phenotype? I don't actually know. I would, we didn't look for that. That data is available because for the 12,000 strains in that data collection, there are whole genome sequences, and there is a collection of SNPs. But we did not specifically look at the other examples of isoniazid resistance. But clinically, like the vast majority of the mutation of catalase.
Yeah,
So-
but to get to the mechanisms, it would be useful to,
Right.
like.
Yeah, but
So the statement is that, like, it would be useful to know if that's true for, those mechanisms would be useful to know. And my proposition is that I don't think that this mechanism is every single clinical strain that has isoniazid. And in fact, the distributions that Maddie was referring to already show you that not all NBR strains behave the same as other NBR strains, and not all drug susceptible strains behave as other drug susceptible strains.
Jason, the HPC mutation that are in the promoter, I mean, they are compensatory when you have that gene mutation.
Okay, so.
Yeah.
So.
The other,
Right.
the other mechanism of isoniazid resistance is mutation in the promoter of INHA, MAR-A, and INHA2.
So Hassan is, bringing up that, like 90% of like isoniazid resistance is like 70+% catalase, and the majority of the other clinical mechanisms of resistance is a change in the promoter that drives expression of the target of isoniazid, which is INHA.
There are mutants described in alkaline hydroperoxidase that are the cause of resistance to isoniazid.
So Marcel is saying that there exists mutations in AHP that can also cause resistance. But I think that they're exceedingly rare, although physiologically they can exist, I guess. Jean?
Well, I was just questioning the use of clinical strains, because each clinical strain is gonna have multiple different mutations. So you don't know what's due to what. So the meaningful experiments, I think, are with your H37RB, and your mutation in CAPG in that strain that's made in the lab. That you know is only that one mutation, and when you complement it works. Because the clinical strains are a complete zoo of all kinds of mutations. So even, as was pointed out, the INH resistance is due to different reasons, but there's other mutations that could cause resistance or sensitivity to
So the comment here is that clinical strains are genomically incredibly diverse and a lot of things, and therefore may make it is that it could be a barrier towards getting to molecular mechanisms if you study the clinical strains. And so what I did not specify, and I should have clarified, is that every single one of these phenotypes that we saw in clinical strains, we saw the exact same phenotype in the laboratory strains.
So I know
And
those
so-
meetings.
Right, and then all these clinical strains is validating that this is actually meaningful in the real world.
Yeah.
Because like more than 70% of the resistance is actually by this mechanism. Which is paper didn't get published because of I think that the clinical work was really important for the editor reviewers. Yeah, I mean, but I guess the other thing is that it's still more than 70% of mutations. So if you have a lot of different things, a lot of observations, and at the mean level, right, you can make some. So just like when we actually did an experiment with, say, a wild type and a mutant strain, even in those experiments, there's a tremendous amount of cell variability. Right? No individual bacteria are exactly the same. Well, right? So it's similar to saying that there are going to be slight differences in physiology between cells in the same culture.
Yeah, but that's not going to affect the PBT resistance. I mean, you're looking at the whole culture. Right? You're not looking at individual cells.
So the comment is that, that these phenotypes are already at the bulk level, so it's not, this is not a single cell level. I completely agree.
Are you surprised that you compared this curve with only the isoniazid units, that the normalized values are identical when you strip away the RIF resistance?
Can you say that again?
So
I missed that last time.
you had a pair of figures
Mhm,
with this, which is comparing MDR with non-MDR.
mhm,
And then you had exactly the same kind of histogram
mhm.
with only isoniazid resistance and sensitivity.
Mhm.
And the curves are almost superimposable and they're normal.
Mhm.
So wouldn't you expect that if I'm trying to figure it out that, I mean, the effect is due, I believe the effect is you're showing an effect for isoniazid resistance, but curve on the left, I guess it's okay.
So if I
I
understand
mean,
the.
if you did exactly the same experiment with RIF resistance strains,
Mhm.
right, there would be no effect. But you have that data.
So David now is asking, these curves look almost identical. I can tell you they're not identical. These curves look almost identical.
They're almost.
So, so I guess the question is, what would you expect for rifaximin resistant? Like when this, could that alternatively explain all the phenotypes or something? What I'm not showing you here is that there's not this clear separation between the RIF resistant, between strains that are RIF resistant and strains that are RIF sensitive. Okay. So therefore, we reason that,
Okay.
because we see this. And then that last experiment with the folate synthesis inhibitors shows you that there are differences in physiology, right, between strains that possess that do not possess a RIF resistance mutation. So those are the isoniazid resistant only, right, versus the ones which possess both, right? So that red one, versus, so thank you guys for your attention. Thank you all, people.